AS Level Biology 9700, Paper 2, Variant 1, May June 2025. And this is the last video on this paper and including question number 5 and 6. So, question number 5 is phosphate ions are absorbed from the soil solution by roots and are needed for cellular processes throughout plants. Scientists investigated the movement of phosphate ions in flowering plants. The scientists discovered that phosphate ions in the leaves are transported from the roots in the xylem. Only a small proportion of the phosphate ions that are absorbed are transported to the growing points of the roots and shoots. Suggest why only a small proportion of the absorbed phosphate ions are transported to the growing points. Before writing, I just like to discuss that why it happens that only a small proportion of the absorbed phosphate ions are transported to the growing points. So the reason is that the phosphate ions are essential for all metabolically active sites, not just the growing points. So phosphate is required throughout the entire plant for synthesis of ATP, phospholipids. Phospholipids, you know the part of the cell membrane and the backbone of DNA and RNA. Phosphate is required. So in this way, the phosphate is requiring everywhere. It is required everywhere. So while growing points have a high rate of phosphate demand due to rapid cell division, the total quantity required by the rest of the existing mature and metabolically active plant bodies is significantly larger, meaning most of the absorbed phosphate is utilized before reaching the growing points. So it is required everywhere. So that is the reason it is used up when it is absorbed and transporting towards the growing points. On the way, it is captured by the other parts which need these phosphate ions. So I would like to write all these points over here. We need 2 points specifically. So phosphate ions are transported for all metabolic active cells, not just the growing points. They are required throughout the entire plant for synthesis of ATP, phospholipids, phospholipids, and the backbone of DNA and RNA. With pentose sugar, it makes the backbone. Growing points, growing points also need hydrate them due to cell division. But most of phosphatides ions are utilized before reaching to growing points. Due to their high demands everywhere. So this can be the reason. This is my suggestion. Now the next part B is Gossypium hirsutum is the most common species of plant grown for the production of cotton across the world. So scientists carried out an investigation to trace the pathway taken by the phosphatines from the leaves of cotton plants into the stems. The scientists used a radioactive isotope of phosphorus to trace the pathway of the phosphate ions. So some cotton plants were divided into two groups, A and B. So in the A group, scientists inserted impermeable waxed paper between the xylem and the phloem. So these are the waxed papers between the xylem and the phloem. So these waxed papers between these in the stem below a leaf of each plant. So below a leaf. So this is the leaf origination. This is node. Leaf is originating, and below it they have inserted the waxed paper. And on the other hand, inserted a solution containing phosphate ions labeled with 32P into a vein of each leaf. So into the vein of each leaf. So this is the wax paper. So it is A group, and so this is the B group. So moving towards the next page. The procedure was required on the plant group B without inserting the waxed paper. No inserting of waxed paper was there. So the procedure was repeated on the plants. After 1 hour, the scientists determined the percentage of labeled phosphate ions in the 4 sections of the stem S1 to S4. See, the results are shown in table 5.1, and you can see in 5.1 the group A stems with waxed paper or group B with stem with no waxed papers. S1, phloem and xylem, you can see both values for them, percentage of injected labeled phosphate ions over there. Now, question is, by keeping in mind this table, we have to go with the answer. Question is a little bit in this way. Yeah. The group A and group B. Using figure 5.1 and the data in table 5.1 to discuss the pathway taken by the solution containing phosphate ions labeled with 32P in cotton plants. So before writing, I would like to discuss that the pathway taken by the solution containing phosphate ions according to this data shows that phosphate is primarily translocated in the phloem from the source leaf with the evidence of lateral transfer into the xylem along the stem. So from the primary translocation in phloem, the percentage of phosphate 32 in the phloem is consistently much higher than in the xylem for all sections in both groups. You can see that in phloem, it is higher as comparison to xylem. It is higher in phloem as comparison to xylem. So it means that translocation or it takes place by the phloem. So in S1, the phloem 12% in uh, A and 15% in group B. So in in xylem it is only 1%, and without wax paper it is 5. So this confirmation phloem is the main route for transport away from the source leaf. So phloem uploading, according to this, the concentration in the phloem decreases from S1 to S4, so indicating continuous uploading of phosphate into the surrounding zinc tissues of the stem as it moves down. So the percentage in the xylem is significantly higher in group B than in group A. So according to this, the wax paper prevents transfer. So the higher reading in group B's xylem proves that the phosphate is transferred laterally from the high concentration phloem into the xylem for recirculation. So I think I have told you that the phloem, it is translocated by the phloem cells, but then you know that the waxed paper is there, which will prevent the entry of these phosphorus 32 into the xylem. But in this case, where there is no waxed paper, the because it is laterally transferred to the from the phloem to the xylem. So some quantity of it is, and so this is the reason the values in the xylem is higher than the xylem with the waxed papers, where there is blockage and there is no entry of P32. So I'm moving towards the writing answer. So I would like to start right from here. The P32. This is the which was injected. So here I would start. In both groups. So you can see the S1 and S2, all they have the higher uh, concentration in the phloem instead of, so the concentration in the phloem decreases from the concentration from S1 to S4, which confirms that phloem is main route for transport away from the source. source leaf. Decrease from S1 to S4 shows the continuous uploading of phosphate in surrounding sinks. Sink tissues the percentage of xylem is higher than group A because of absence of waxed papers. Which allows the transfer laterally from the from phloem to xylem. Next question, number 6. And question number 6 is figure 6.1 is a ribbon model of a molecule of hemoglobin. So you can see they have labeled one part with X. State the part of the hemoglobin molecule labeled X. So this is the labeled part X. So according to the structure of hemoglobin, the hemoglobin having quaternary structure, which is meant by, let me explain. Hemoglobin has quaternary structure. Which is three-dimensional structure. Three-dimensional arrangement of 4 chains, 2 alpha and 2 beta. Each has a heme group. Each chain has a one heme group. So this X is the heme group. And what is heme group? Oxygen always binds with it. to make oxyhemoglobin. So state the function of the structure labeled X. The function of the heme group is to bind with the and transport oxygen. To bind and to bind with and transport oxygen. Very simple questions. Moving towards the question part 3. Hemoglobin is described as having quaternary. So what is meant by quaternary structure? Already that is the quaternary structure. Specific quaternary structure is the specific three three-dimensional arrangement of two or more separate two or more separate polypeptide chains to form a single protein single functional protein molecule. So next question, let me read, then we will move. The effect of the partial pressure of oxygen and the effect of the partial pressure of carbon dioxide on the percentage saturation of hemoglobin was investigated. So sample of mammalian blood was exposed to a gas mixture that contained increasing uh, partial pressure of oxygen. In the experiment, the uh, partial pressure of carbon dioxide was maintained at 2.7 kilopascals. The percentage saturation of the hemoglobin in the blood sample was determined as the partial pressure of oxygen increased. So the experiment was repeated with further samples of blood with partial pressure of carbon dioxide maintained at 5.3 kilopascals and the at 10.7 kilopascals. So the results are shown in figure 6.2 and 6.2 has been shown over here. And this graph shows the three curves. This graph shows the three curves as, let me move this page. Yes. So you can see the three curves over here on the graph, which I should add. First of all, the partial pressure 2.7 kilopascals, and then which is this curve. And second curve, which shows the dotted line, so 5.3, and very small dots, it shows 10.7 kilopascals. So percentage saturation of hemoglobin with oxygen, and so these are the three curves which has been shown over here. So we will move what they have asked first, then we will come back to these curves. So the partial pressure of air, alveolar air is 5.3 kilopascal. State the likely partial pressure of oxygen in the alveoli of mammal. So this is called as the partial pressure in the blood leaving the lungs at the same as the alveolar pressure. So diffusion results in equilibrium. Therefore, the leaving the lungs is about 13.3. It will be nearly equal. So it will be something range between 12 to 14. So it can be between 12 to 14. So these. So it is the equal. It is in equilibrium. So we can say that it is between 12 to 14. We can write a range over here. We can write this range over here. That is, it is between 12 to 14. So moving towards the next, suggest the range of partial pressures of oxygen in respiring tissues and use figure 6.2 to give evidence for your answer. So first of all, I would like to move towards this this uh, this graph, which is 6.2. So we have to discuss firstly this. You can see over here we have to find out the range. The curve from respiring tissues is the one with the highest partial pressure of carbon dioxide. So you can see the highest pressure of partial pressure of carbon dioxide is there, which is 10.7 kilopascals. So it is with small dotted line representing. So this shift to the, you can see the curve has been shifted towards the right. So this shifting to the right and down is known as the Bohr effect. This is called as Bohr effect. So respiring tissues are, why it happens, that is the respiring tissues are metabolically active and produce a significant amount of carbon dioxide. Uh, so as a waste product, this carbon dioxide is produced. So this quickly dissolves in the blood, increasing the partial pressure of carbon dioxide. So and through its reaction with water, lowering the blood's pH. So this condition in respiring tissues causes a change in the hemoglobin molecule structure, which reduces its affinity for oxygen because carbon dioxide concentration is high over there. So this reduced affinity is graphically represented by a right shift and downward shift of the oxygen hemoglobin dissociation curve. You can see. So this curve has been shifted to this towards the right. So it has been shifted. So this one. So the right shift means that for any given partial pressure of oxygen, the percentage saturation of hemoglobin with oxygen is lower. In other words, hemoglobin is more like to release its oxygen into the surrounding situation. Therefore, the curve representing the conditions in respiring tissues will be one with the highest carbon dioxide, which is the dotted line. So the other two curves, particularly the solid time, represent conditions closer to the lungs, and this one, the closer to the lungs, because very lowest concentration of carbon dioxide, and the curve that is from, and this is from respiring tissues, and shifting towards right shows the Bohr effect. So you can see respiring tissues are metabolically active. So in this way, the shifting of the work takes place towards the right. So move towards the question after discussion on graph. I would like to move. So this you can see the steepness, this steepness you can say till 6. The steepness is till 6, shows the, from the graph you can see. So I would like to write the, that the range is between it was between 12 to 14. Now it is between 0 to 6 or 1 to 6. You can write any of this. That is between 1 to 6. And I have shown you the steepness of this on the graph. The curve with highest concentration of carbon dioxide is shifted to the rightward, which shows this means that any partial pressure of oxygen, the hemoglobin saturation is lower. Means releasing oxygen. More ready, because the concentration of carbon dioxide is its peak over there in the respiring tissues, and here the carbon dioxide is producing as a waste product. So the here I'm moving towards the next question. The question is, so I would try to write this answer that use by using the figure 6.2 effect of increasing, as already I have discussed the answer into the previous question, but I would like to write right now as partial pressure of carbon dioxide increases. You can see it is highest in the lowest curve, which has been shifted to the right. So increases the oxygen hemoglobin dissociation curve shifts to the right. Shifts to the right and down, indicating a reduced affinity of hemoglobin for oxygen and thus a lower percentage saturation. Saturation for the same partial pressure of oxygen. This change facilitates the unloading of oxygen in respiring tissues. So next question is, state the name given to the effect you have described in part 3. So the name given to the effect where an increase in partial pressure carbon dioxide causes a reduction in hemoglobin affinity for oxygen is called as the Bohr effect. The last state, the last question is, explain the advantage to the mammals uh, effect you described in part 3. So I am going to this answer that what can be the advantage? The advantage of the Bohr effect is that it enhances the unloading of oxygen where it is most needed. Firstly, I would like to first advantage. The advantage of the Bohr effect. Bohr effect means right shift of the curve at high carbon dioxide concentration is that it enhances that it enhances the unloading of oxygen where it is needed. where it is most needed. This lower and higher carbon dioxide reduces the affinity of hemoglobin for oxygen. So this hemoglobin gives up its oxygen more rapidly. The hemoglobin gives up its oxygen more rapidly at the same at the same oxygen, partial pressure of oxygen compared to the lungs. Maximizing the release and diffusion of oxygen into the tissues with the highest metabolic demand. So students, it was all about this paper 2 variant 1, and in the next video inshallah we will start paper 2 variant 2. Don't forget to like, share and subscribe my channel, because in this way you will be able to watch more papers, to study more papers, to understand more papers. So please don't forget to like, share and subscribe my channel. Thanks for watching.